Hello everyone, myself Ms.M.Vishalatchi, Assistant Professor, Department of Botany. So today we are going to see about linkage. So linkage was discovered by William Batson and Punnett. They have worked in Lathyrus odoratus, but the experimental result was given by T.H. Morgan. He worked in Drosophila melanogaster. First we shall see the definition of linkage. Linkage is defined as the tendency of two or more genes to remain together in the original combination in the same chromosome during the process of inheritance for a number of generations. In the image you can see the gene 1 as well as the gene 2 both are remain together and they will remain same in the process of inheritance for a number of generations too. So now we are going to see about the arrangement of the linked genes. So we know in genes there are two types, one is dominant allele and one is recessive allele. When both the dominant alleles are present in the same gene, that is known as cis arrangement. When the two dominant genes are present on different chromosome, that is known as trans arrangement. So next we are going to see about the theories of linkage. So there are two types of theory, one is differential theory, then the other one is chromosomal theory. So first we are going to see differential multiplication theory. In differential theory it was proposed by Batson in 1930. He assumed that the differential multiplication of cells containing different combinations of genes is responsible for linkage. But this theory was disproved because after segregation of gametes there is only one division. So next the chromosomal theory, it was put forward by Morgan and Castle. So according to their theory, they have the postulates that the genes are arranged in a linear fashion on the chromosome, the genes on the chromosome are linked, the genes showing linkage are located on the same chromosome, they always remain together, and during the process of inheritance, the distance between the genes determine the strength of the linkage. So in the linkage there are two types, one is complete linkage and the other one is incomplete linkage. The main difference between these two is in complete linkage there will be the absence of crossing over, in incomplete linkage there will be the presence of crossing over. So there are a few factors which affects the linkage, mainly the distance. If the genes are present close together, they show strong linkage. If they are distant apart, they show weak linkage. The age, temperature and X-rays are always having a decreasing effect. When the age increases, the strength of linkage decreases. When the temperature increases, the strength of linkage decreases, as well as when the X-ray treatment increases, the strength of linkage decreases. So what are the significance of linkage is that linkage presents variation in evolution. It plays an important role in determining the nature and scope of hybridization. It reduces the chances of formation of new combination of genes in gametes. It helps the parental, racial as well as the specific trait to be together. These are the significance of linkage. So today we have seen about an introduction class about linkage. Thank you all.